స్లైడ్స్ అనేవి మీరు బాగా స్టార్ట్ చేయగలరు. ఇది మార్చు డెఫినేషన్. మరి ఇవి స్లైడ్స్ కూడా చాలా
వస్తుంది.
Hello. Am I audible? Hello.
Sir, is it audible, sir?
Am I audible, madam? Hello.
Sir, good morning. How are you, sir?
Good morning.
Good morning, sir.
Am I audible properly?
How are you, sir?
Good morning. Good morning. Am I audible
I
properly?
hope everything is fine over there.
Fine, sir. Fine.
Okay, sir.
Yes, yes, sir. Yes.
Another 5 minutes, we will start the session, sir.
Okay.
You are ready, sir?
Sure, sure. I am ready. Yes, yes. I
Yeah,
am ready. I
yeah.
am ready. So
Yes.
shall I share the screen?
Just,
Okay.
yeah, you can share this.
Madam is coming, sir. Speaker, sir. Sir, could you please share your screen, sir?
Yes, yes. Is it visible, madam?
No, sir.
Is it visible?
No, sir.
I have shared already, madam.
Okay, sir.
Visible, madam? I have shared the screen.
Screen share. Shweta? Sir, call is supposed. Sir, your screen is not visible, sir.
Yes, yes, madam. I am trying. Is it visible, madam, now?
No, sir.
Now?
No, sir.
Now, in my screen, it is already shown that this is you are sharing your screen on Zoom. It is showing, madam. But why? From your side,
Okay, sir.
now visible?
Yes, sir.
Okay, fine.
Okay. Good morning, everyone. It is my great pleasure to welcome you all to this morning session. I extend a warm welcome to our respected FDP conveners, Principal Professor K. Tejamurthy sir and Dr. Mastanama madam, our keynote speaker, Dr. Pintu Bhagavanbhai Prajapati, faculty members, and dear participants. We are truly honored to have such an esteemed gathering today. This session promises to be insightful and enriching for all of us. Now it is my privilege to invite our keynote speaker for the session. Before I invite our keynote speaker to deliver the address, let me take a moment to briefly introduce our keynote speaker. Dr. Pintu Bhagwanbhai Prajapati is an associate professor in Maliba Pharmacy College under UKA Tarzia University, Bardoli. Sar has awarded PhD in the year 2014 under the University Veer Namrat South Gujarat University. Sar has an experience of about 18 years of academic experience and 16 years of research experience, including quality assurance and quality control, formulation development and characterization, stability study of API and pharmaceutical dosage forms, analytical method development and validation, herbal drug analysis, biosensing and bioimaging. And also currently SAR is guiding 7 research scholars, one of them are awarded PhD. SAR has published 5 national journal publications and 95 international journal publications, which are having 175 plus impact factors. SAR has granted a research of 12 lakhs from different government funding agencies. SAR has awarded 3 times Dr. P.D. Sethi Memorial Annual National Award in the year 2020, 2022, and 2023. SAR is a lifetime APTI member. Currently, Dr. Pintu Bhagwanbhai Prajapati Garu is working as an associate professor in Malibu Pharmacy College under UKA, Tazia University, Bardoli. We are truly privileged to have such an eminent personality with us today. On behalf of ANU CPS, it is my privilege to invite our keynote speaker, Dr. Pintu Bhagwanbhai Prajapati Garu, to address the gathering. Thank you, sir.
Thank you, madam. Thank you for the nice introduction. Am I audible properly?
Yes, sir.
Yes, sir.
Okay, and the screen is properly visible?
Yes, sir.
Yes, sir.
Okay, okay. So first of all, I am thankful to Dr. Tejamurthy sir for giving me opportunity as a resource person in this 1 week FDP on next generation drug discovery, and also thankful to the management, Acharya Nagarjuna University, for giving me opportunity as a resource person. Okay, so first of all, we have to discuss the background why this particular topic is selected and how it is. We can say that according to the whatever the theme is selected for the FDP, right, which is dedicated to the development of analytical method by the implementation of quality by design. So if we're thinking about the regulatory perspective, so now onwards, when you are going for US FDA filing, okay, so in that case, the analytical method should be developed by the implementation of the analytical quality by design approach. That's why this topic is selected for the application for of new drug substance, new drug product. So today we will see how the quality by design approach can be implemented for development of any analytical method. Okay, it may be HPLC, it may be HPTLC, or it may be any other analytical method in which how you can go for step-by-step implementation of quality by design approach that we will see today. Okay, so it will be according to the guidance given in ICH Q14. Okay, so according to that, we will discuss each and every steps how you can proceed for the implementation of this approach. Okay, so as per the contents, we can say first of all we will see what is the analytical quality by design, then what are the regulatory requirements for the AQPD approach. Obvious, there are already some ICH guideline which is supporting the QPD approach, ICH guideline number Q8, 9, and 10. But these guidelines are dedicated for development of the process and product for manufacturing of new drug substance and products. It is not dedicated guideline for the analytical methods. So certain terminologies which are used for the ICH guideline number Q8 is not suitable for analytical quality by design. That's why we will see some terminologies how they are differ from the QBD approach, which is mentioned in ICH guideline number Q8. Then we will see what are the steps. How can you go for step-by-step implementation of QBD approach? Then we will see case study, published paper. We will take as a case study, and we will see how we implemented that approach to the development of RPHPLC method. Okay. Now we are starting with the analytical quality by design approach. So as per the definition of the QPD approach, we have derived the definition of the analytical quality by design. So the analytical quality by design approach is the systematic approach to the development of analytical objectives and give more emphasis on behavior understanding and instant results as per the predefined specifications and intended use of the analytical method using principles of quality risk management and design of experiments. Okay, so here our main emphasis when you are going for implementation of quality by design approach to the development of any analytical methods. So our main aim when we focus on the development of analytical method, so it is always result oriented. So what are results we are getting? So results are controlled by the analytical method variables, analytical method parameters. So here we have to start the approach step by step, so we can obtain the proper understanding for the understanding of the that behavior of the method. Then we can obtain the control over them, and we control those parameters in such a way that we will get the results of the analytical method as per our desirability, okay, as per the regulatory requirement. Okay, and for this we will take help of some software, some tools. These are the quality risk management and design of experiments. Okay, so this is the definition of the quality by design approach. So here I have mentioned that predefined specifications. So always remember that whenever you start the analytical methods, it is always starting with predefined specifications. So here QBD approach is also emphasis that you have to have clear predefined specification before you starting the development of any analytical method. So we'll take the example of HPLC. Then when you start the HPLC, then there should be a proper predefined specifications you are taking from the pharmacopoeia. So pharmacopoeia is mentioning system suitability testing parameters for the resolution of the peak, tailing factor of the peak, capacity factor, number of theoretical plates, etc. So as we know that the pharmacopoeia is recommending that the resolution should be more than 2, tailing factor range of 1 to 10, then number of theoretical plates should be more than 2000. So these are the already the acceptance criteria given in the pharmacopoeia, and keeping this particular target in our mind, we will start the development of analytical method. Okay, and obvious we also recommending that the whatever method you are developing, it should be properly validated as per the ICH guideline number Q to R2. Different parameters, validation parameters are mentioned there, like accuracy, robustness, reproducibility, precision. Okay, so according to that, the guideline should, whatever analytical method you develop should be properly validated. And don't forget what types of analytical method you are going to develop. It should be clear. Okay, so that will decide the application of analytical method. So are you going to develop the RPHPLC method for stability study, or for assay, or for dissolution, or for bioanalytical purpose? Okay, so number of results are there, number of applications are there. According to that, you can select the types of analytical method. So there should be your predefined specification before you start any development of analytical method using quality by design approach. Okay, so here that is the main source for the predefined specification should be a pharmacopoeia, or it may be a ICH guideline. From that you have to take the specification and acceptance criteria for those parameters you are setting for predefined specifications. Okay, now, before starting the approach discussion, first of all, we have to understand what benefits we are getting out of this. Okay, so right now, most of the are practicing the development analytical method by a conventional approach. That is one factor analytical method by implementation of quality by design. Then you will get the number of advantages, like you can assure about the robustness, accuracy, and precision of the analytical method. So more, we can say that high level of the validation parameters results you will get if you develop your analytical method by implementation of QPT approach. Obvious now ICH guideline Q14 is in public domain, so it is a one of the regulatory requirement for submission of application. Then obvious post-approval change management is somewhat become flexible if you go for the quality by design approach implementation in development of analytical method. So here, if you go for one factor at a time, so during the life cycle management of the analytical method, you need more frequent post approval change management. But when you develop that analytical method by implementation of QPD approach, then we can say that very less chances are there you are getting any change out of design. So out of design space is only considered as a change, and within design space, if you are changing any parameter, then it is not considered as a change. So there is a, we can say that high level of the flexibility we are getting for the post approval change management system. So your documentation task will be less hectic when you go for QBD approach to the analytical method development. Obvious, one factor at a time, you going for two dimension X and Y, you are not thinking about the third dimension. So interaction is basic event approach. So here you can explore the interaction between two factors, which is very easy by the QBD approach implementation to the analytical method. So that is the major advantage we are getting here for the implementation of QBD approach to the development of analytical method. Here you can run two parameter at a time, and you can study the interaction between two parameters for getting the maximum results as per your desirability. So that is the major advantage you can explore here by the implementation of QPT approach. Obvious, it is helpful for obtaining the process control and capability in SPC. So it is very easy here if you go for QPT approach. List documentation is required because there is a less task is available for the post-approval change management. So very list documentation is required when you go for QBD approval. Life cycle management is very easy approach. So one factor at a time, it will be taking more life cycle management little bit easy when you go for QBD approach. Okay. Now, next we will see the terminologies. What are the different terminologies that we have to study before starting the QBD approach? Terminologies are little QBD approach, which is mentioned in ICH guideline number Q8. Based approach, which is already they are emphasis that how we can go for step-by-step implementation of QBD approach, so that the risk management of the analytical method will be easy. Okay, so that is ICH guideline number Q14. This terminology is, we can say, a little bit different from the QBD approach what you apply as per the ICH guideline number Q8. Here in ICH guideline number Q8, you are talking about the quality target product profile, but instead of quality target product profile, we have to use the terminology that is analytical target profile, or you can also use quality target analytical profile, or you can say quality target method profile. So these are the terminologies you can use interchangeably with the QTPP when you go for QBD approach. Profile. Now, what is the analytical target profile? It is a prospective summary of the quality characteristics for the results of the analytical method, which assure its predefined specifications and a regulatory perspective and its application. So, whatever quality characteristics you desired from the performance of the analytical method, that should be listed in analytical target profile. So along with analytical target profile, you can also include the quality target product profile for what product you are applying that analytical method. So that can be integrated with this one. So many research paper you can see QTPP with ATP. Okay. Next one is the terminology we are using here, that is CQA you are using in Q8. In here you can use CAA, or you can say the AQA. Whatever. So that is called critical analytical attributes, or analytical quality attributes, we can say that. So these are the acceptance criteria for development of analytical method as per the pharmacopoeia, as per the ICH guideline. What should be acceptable, what should not be acceptable. So these all parameters you can trace from your analytical target profile, and you can make the diagram. Ishikawa diagram is a, so here the presentation of the all analytical method parameters is very easy, so that you can focus one by one to each parameter, okay, without missing any important one. Then next step is ICH guideline number
ఆఫీసులు మొన్న కొట్టారు
ICH guideline number
ఆఫీసులు
Q9 is
ముందుగానే కొట్టారు
helping here,
అది
that is
విత్డ్రా
analytical
చేయండి అది మొన్న రాసి అది సెలవులో పెట్టుకోవాలి
Sir,
సెకండ్
please
సాటర్డే
mute
సండే
other
assessment of
participants.
the risk
ఫోన్ చేశాను
assessment
కదా ఈ రోజు వస్తే
assessment of the
తీసుకొని
risk.
వస్తాను అక్కడికి పోతాను
Hello, hello. Is there any issue?
ఫోన్ చేశాడు
Am I audible properly?
హలో హలో
Hello. Am I audible properly, sir?
Sir, you are audible, sir. Please mute other participants, sir.
Okay.
Green.
So here, once you make the list in Ishikawa diagram, you fix the list in Ishikawa diagram, then we have to check each and every parameters for its risk to the results of the analytical method. For that, the ICH guideline number
Sir,
Q9
please
is
mute other
helping here.
participants.
Yes, admin, please mute the participants so that it will be easy to convey the message. Okay, now it is okay. Now we can continue. So risk management is one tool as per the guidance given in ICH guideline number Q9. Each parameter what you mention in the Ishikawa diagram should be tested its risk to the development of analytical method. From that, you will get the list of the critical method parameter which are very very risky to the results of the
Madam,
analytical method.
ఆ
Means
రెండు
if
బిల్లు
you
సార్ సైన్
can
పెట్టి
say
ఫిక్స్ చేసింది
these
మేడం.
parameters,
ఆఫీస్
if,
కావాల్సిన ఆఫీస్ ఖరాబ్ పెట్టు పెట్టేసాను ఫిక్స్ చేసి పెట్టేసాను
I think there is some
రేపు
background
వచ్చినప్పుడు
noise
పెట్టేస్తే
I am getting.
మళ్ళీ ఆయన ఉంటాడు ఉండడా రెండు బిల్లు ఈ కవర్లో పెట్టా దీంట్లో రెండు ఇచ్చేసేయండి
Sir, please
వచ్చినప్పుడు
mute
తర్వాత
Hello.
others.
Please mute yourself. Please mute yourself. There is a
As a
అది మన
host,
దగ్గర
you can
large
mute,
పెట్టుకున్నాడు
sir.
background
Please
noise
mute all the
coming in.
participants.
అది మన దగ్గర పెట్టుకున్నాను చూడండి లిస్ట్ లో రాశారా
Kindly mute yourself.
ఆ లిస్ట్ లో కాదు మనకి బుక్స్ బడ్జెట్ చేస్తుంది ఎదిగండి సరే
Hello.
రెండు సెట్లు ఉన్నాయా
Sir, please unmute your mic, sir. Sir, please unmute your mic. Sir, your voice is not audible, so please unmute your mic, sir.
Now am I audible?
Yes, sir, you are audible now, sir.
Am I audible?
Yes, sir, yes, sir.
Okay. Is the screen visible?
Yes, sir, everything is clear, sir.
Okay, fine. Okay, so how we are getting the critical process parameters here? We are getting the list of critical method parameters. These are the parameters which are giving directly impact on results of analytical method, so that you can identify by the risk management. Then these critical parameters should be properly controlled. So for that, you can use the tool that is design of experiments. Now, what is design of experiments? Design of experiment is a process to link the critical process parameters, critical method parameters to the critical analytical attributes to get the desirable results. Okay, so for that you can take the help of DOE, and you can understand the behavior of the critical method parameters. And once the behavior is properly understood, then we can obtain the control over the that parameters, and that control is resulting into the desirable region that is known as analytical design space. Okay, so here we are getting the analytical design space instead of design space, or other terminology which can be used, that is analytical, we can say that method operable design region. Okay, so MODR is also proper word we can use here to represent the design space in analytical quality by design. So these are the different terminologies that you should know before you are starting the implementation of QPD approach to the development of analytical method. Now we will see what are the steps. So first of all, we will see step by step how can you proceed for implementation of QPD approach. So first of all, you have to clear ATP or QTAP for analytical method. In here, we have to include all predefined specifications, all applications, everything should be included in ATP for the proposed analytical method. Okay. Once the analytical method is decided, then you can go for composition of analytical along with PCAs. You can go for composition of Ishikawa diagram, cause effect diagram, or fishbone diagram. And after that, one by one, you can go for step number, that is analytical risk assessment of each and every parameter. And from that, you will get the list of the critical method parameters, and critical parameters, method parameters are huge in number, more than 4 or more than 5. Then you can go for DOE. The first step of DOE you can try, that is screening design. Okay, so screening design is required when there is a more than 4 parameters you are getting after the risk assessment which are critical for the method. Then if you are getting less than 4, then you can directly go for response surface analysis in the DOE. And after that, you can get the step number 5 by overlapping the whatever 2D or 3D contour plots from the DOE. After overlapping, you can get the desirable region, that is called MODR, method operable design region. And from that you can obtain your control strategy for the life cycle management of analytical method. Then whatever design space you are getting, that design space should be validated for its performance. So whatever predicted results you are getting from the software, that predicted results should be tried in the laboratory, and after that it should be compared with the actual results. The percentage RST should be less than 5% between predicted and actual. Then you can say the model is properly validated. Then obvious you will get one particular operating condition from that design space and control strategy, and that will be your optimized chromatographic condition. And once you get the optimized chromatographic condition, then you can proceed for validation as per the ICS guideline number Q2R2. And after the validation, you can apply the analytical method for the whatever purpose you developed it. Okay, so different samples are there. According to that, whatever sample you are targeting for the application of the analytical method, that particular sample you have to apply. Okay, once validation is over. So these are the development steps and validation steps. You proceed for the development of the analytical method by implementation of quality by design approach. Here I have already told you that the step number one, in step number one, you include the intended use, predefined specifications, list of potential CAs, list of potential method parameters. Everything should be included in this step. Okay, once it is fixed, then you can go for Ishikawa diagram. Okay, so in Ishikawa diagram, you can categorize all the parameters properly, and you can study all the parameters by the risk assessment. So step number 3 is the risk factors according to its severity, occurrence, and detectability as given guidance in ICS guideline number Q9, and go for multiplication of that score, and you are getting the risk priority number, and that risk priority number you can classify for the high risk, low risk, and medium risk parameters. And high risk parameters you considered as a critical one, and these critical parameters should be carried forward for the optimization. Okay, so in optimization, step number 4, here you are having two options. Either you can go for screening design first, then response surface modeling, or you can directly go for response surface modeling. And for that, I have given some examples. Suppose you are getting more than 11 parameters which are to be critical, then you can try the placket Berman design in DOE. And suppose you are getting the parameters in the list of 7 to 11, then you can proceed with the Taguchi orthogonal array design. If you getting the parameters in the range of 4 to 7, then you can select fractional factorial design. And if you are getting less than 4, then there are a number of options are available, like box Banker design, full factorial design, central composite design, mixture design. So these are the different options what you can try for whatever number of parameters you are getting after the risk assessment. Okay, so according to that, you can go for DOE for statistical analysis and graphical relationship between the critical method parameters and critical analytical attributes. Okay, so this is the step number 4. Here you can take the help of the design of experiments. So design of experiments will finally give you the desirable region. So that desirable region is your method operable design region. You will get the 2D and 3D contour plots, and after overlapping the 2D and 3D contour plots, you will get the MODR, and that MODR will be validated, and according to that, the control strategy should be framed. Okay, so from that control strategy, you will get any one optimized condition, and finally you are getting the whatever results you targeted in that ATP. So this is the outcome of the analytical quality by design. Finally you can proceed for validation. After validation, you can apply your targeted method for the sample. Okay, now here we see the case study what we selected for HPLC method development, QBD approach implementation. So this paper is already published in Journal of AOSC International. Okay, that is Oxford University Journal. So here the title of the case study is DOE based analytical failure mode critical effect analysis. So here DOE is the tool what we using, and analytical failure mode critical effect analysis is the risk assessment tool we are using. So both risk assessment and DOE is mentioned here to multi-purpose RPHPLC method for estimation of multiple fixed dose combination products of metformin using analytical quality by design approach. So here our sample are multiple fixed dose combination of the metformin. As you know that metformin is the most combinations are available in the market along with the metformin. Okay, so here we have go through the literature, and we found that multiple RPHPLC method are available for assay of the each combination. So if any say one formulation, then there will be a separate and dedicated RPHPLC system they have to obtain. Okay, so there are the different RPHPLC system is available for each and every combination. Then we tried why we are not going for single chromatographic condition for the estimation of the combinations of metformin. Okay, so here by applying this approach, we minimize the solvent wastage, we save the time. Okay, so we minimize the cost of analytical method. So this is the idea for the selection of the sample, and we chosen the analytical quality by design approach for the development of RPFPLC method. Okay, so this is the work what we carried out in our laboratory. The drug we selected are metformin hydrochloride, pioglitazone, sitagliptin, and gliclazide. So we can say that number of combinations are available here for the 4 drugs. Okay, and first of all, the step number 1, we started here. You can see the multi-purpose RPHPLC method has been developed and validated for synchronous estimation of fixed dose combination of metformin along with pioglitazone, sitagliptin, and gliclazide azide, which should give the resolution. Now we have taken the predefined specification here. Resolution between the peaks should be more than 1.5. Tailing factor of each peak should be less than 1.5. Number of theoretical plates should be more than 2000 as per the pharmacopoeia standards. Okay. And here, what critical analytical attributes we selected? Retention time, resolution, tailing factor, number of theoretical plates were selected as a critical method performance attributes as per the targeted HPLC method. This is the analytical target profile we reported, and according to that, we have made the list of the method variables, method parameters here, and it is categorized in 6M category. And what are the 6M categories? Measurement, method, material, machine, mother nature, and manpower. So these are the 6 category we classified all the parameters here. Okay, and finally we go for risk assessment for each and every parameter according to their severity, occurrence, and detectability, and they are classified. You can see in the graph, red zone is high risk method parameters, medium risk is green, and blue is the least risky parameters. And least risky parameters you can avoid because they are not showing any significant impact. Okay, now from that we found the list of the parameters, that is three. What parameters we are getting here? Percentage acetonitrile in mobile phase, pH of the mobile phase, and flow rate. So these are the three critical method parameters we are getting. And for three parameters, the best design option is box banker design. And here box banker design we selected. And response we are taking, that is the tailing factor and number of theoretical plates. Here metformin is a higher amount in all dosage form, and because of that higher amount of the metformin with the other drugs, there is a major problem with tailing factor of the metformin peak and number of theoretical plates, and that control is required. Okay, so that's why these critical analytical attributes are selected as a response 1 and response 2. So we enter all the parameter in design expert software, and we get the metrics from the software, and we tried each and every parameters, each and every experimental run suggested by this software in laboratory, and we measure the tailing factor of the metformin peak and number of theoretical plates, and all the data through the software and using ANOVA. And finally we are getting the significant main effect, significant interaction, and significant non-linear relationship. So according to that, we are getting the results. And finally we study the 2D counter plot for tailing factor and number of theoretical plates. We are getting quadratic model, which is significant here. And from that we are getting the overlap plot, that is called method operable design region. Okay, so that method operable design region we tried here, and you can see here the yellow shaded region. Yellow shaded region is the region in which if we working, then we will get the proper shape of the metformin peak with tailing factor less than 1.5 and number of theoretical plates more than 2000. Okay, so this region we explored, and it is validated properly by predicting some trials here, and they actually perform in the lab. And whatever results from that, it is compared with the predicted results, and it is calculated for percentage RSD, and we are getting percentage RSD less than 5%. So we can say now whatever design region we explored, that is validated, and that is resulting into the control strategy. The control strategy was framed for the control operating of critical method failure modes, means parameters for the life cycle management of the HPLC. The set of the control strategy said that the multi-purpose RP-HPLC method was developed using ShimPack ODS C18 column, 250 millimeter in length, 4.6 millimeter in diameter, 5 micrometer particle size. That is the stationary we used, and acetonitrile and 0.1 percentage volume by volume triethylamine in water with pH adjustment of 3.2 and ratio of 40:60 be taken, keeping the isocratic flow rate as a 1 ml per minute, and the detection wavelength we carried out, that is 210 nanometers. So this is a control strategy we tried, and this condition is selected for the development, and this is the output we are getting. So we can get the each and every peak in proper shape with proper the whatever pharmacopoeial conditions are there. This is the linearity we are getting. We can see the reproducibility of the results without disturbing the shape of the peak and broadness of the signal. Okay, so these are the output we are getting. So that is the case study we discussed by implementation of QPD approach step by step. Now we'll see some public publications we published as per the QPD approach. So recently published a Journal of Liposome Research. That is the formulation developed by the pharmaceutics department, and we estimated that one by development of the spectropolarimetric method. The spectropolarimetric method is developed by the QBD approach. Second article we get published in luminescence journals. Here we use the risk assessment tool, that is failure mode effect analysis with the design of experiments for microwave aided spectropolarimetric method for estimation of tenalic leptin. Okay, so here we estimated spectropolarimetric estimation of the tenalic leptin by the microwave spectropolarimetric method. The another article we published in that Journal of Fluorescence. Here we use the QBD approach for the development of spectropolarimetric method for estimation of the brucine, and brucine formulation is developed by the physics department. Next article we published, that is pimasertan estimation by Hens reaction. Hens reaction is making the pimasertan fluorescent because pimasertan is self non-fluorescent drug, and for that the reaction is optimized by the QBD approach. Okay, that is getting published in Fluorescence Journal. Next one is Future Journal of Pharmaceutical Sciences. Here we tried the different formulation of metformin, and by the HPTLC method is developed by the QBD approach. Okay. Next is published in Analytical Biochemistry. Here, tenalic leptin loaded solid dispersion adsorbate were studied for the in vivo by the spectropolarimetric method, and spectropolarimetric method was developed by the QBD approach. Next is we are getting publication in Luminescence Journal. Okay, so here spectrophotometric method, Mira background loaded nanostructure that lipid carrier is developed by the physics department, and pharmacokinetic study of this is carried out by the developed spectropolarimetric method. So here the microwave chemical reaction is optimized by the QBD approach. Next paper we are getting in Microchemical Journal. Here we use the NVDCL. NVDCL as a fluorescent agent, and Mira background itself is a non-fluorescent drug. Okay, so here microwave reaction by QBD approach, and HPTLC is the method in which the fluorescent probe is used. Okay, and that is the result we are getting. Next, we published in the Spectrochemica Acta Journal. Here we tried the micro-assisted sensitive reaction, spectropolarimetric reaction for the Mira background. Okay, and here 4-fluoro-7-nitrobenzofuran was used as a biosensing probe. Okay, next one is LCMS method we also developed using green solvents. Okay, so Mira background encapsulated nanostructure lipid carrier were studied by implementation of QBD approach. Next, we developed spectropolarimetric method for ibrutinib loaded nanostructure lipid carrier, and the spectropolarimetric method was developed by QBD approach. Next, we tried the publication in Journal of Separation Science. Here we developed the HPTLC method for estimation of stability indicating estimation of cilnidipine, and we also isolated the whatever degradation products we are getting and characterized, and the development of the method was carried out by QBD approach. Next, we get publication in same journal, that is Journal of Separation Science, HPTLC method for stability indicating estimation of thiocolchicoside and aceclofenac. So that is by QBD approach we developed. Next, we tried that is published in Chemistry Africa Journal. So different anti-diabetic drugs combinations were estimated by RPHPLC method using single chromatographic condition. That's why it is a multi-purpose RPHPLC method, and the whole method is developed by the QBD approach. Next, we tried some chemometrics claims, principal component analysis and DOE. Principal component analysis was applied for risk assessment, and DOE is for optimization. So we developed the RPHPLC method for some combination of anti-hypertensive drugs. So the QPD was the concept. Okay. So AOSC International Journal publication we are getting. Next, we will get the publication in Journal of Chromatographic Science. Here we used to design, that is Taguchi orthogonal array and box banker design we use. So Taguchi orthogonal array is a screening design, And box bunker design is a response surface modeling design here for development of HPDLC method for simultaneous estimation of acyl sartan and silnidipine. So that publication we are getting in Chromatographic Science Journal. Next publication we are getting in Journal of Iranian Chemical Society. Here we use the DMAC principle of 6 sigma here, and we estimated the metronidazole and ofloxacin by HPLC method. Here we tried the QBD approach. So we are getting in Journal of Planar Chromatography. Here we tried the estimation of acyl sartan and chlorothalidone by chromatography. Here we carried out the development by QBD approach. Next, we get the publication in Journal of Pharmaceutical Innovation. Here we tried the multiple combination of anti-diabetic drugs by single chromatographic condition, and that chromatography is developed by the QBD approach. Next, we are getting in planar chromatography. That is, we tried the stability indicating assay of the apremilast, and it's also applied for the degradation kinetics study. Next, we are getting Springer Nature Applied Science Journal. So here we tried the estimation of combination of tizanidine and nimesulide by HPTLC, and HPTLC method was developed by QBT approach. Next, we are getting in same journal for apremilast HPTLC stability indicating method. Next publication means number of publications are there. You can see here. One positive part is there. Some publications are also recognized at national level. So the PDCT Memorial Award is organized every year, and they invite published research paper, and the published research papers are go through the screening with the critical reviewer. Okay, and finally they screen some top three papers, and they give some cash prizes along with the paper. So one paper is getting second prize, that is of 25,000 rupees in year 2022. Okay, so this is the paper, and one another paper is getting a 15,000 cash prize, position in year 2022, and that article was published in Chromatographic Science Journal. Next, recently we are getting that is 25,000 rupees cash prize, second position in year 2023, and that publication we are getting in Journal of AOLC International. So that's all about the QBD approach to the development of analytical method. Hope you will get some understanding related to this. This is my contact detail. If you need any help, any type of guidance related to this, then you can contact. Thank you. Thank you very much for the patience listening. All done from my side, sir. Hello. Hello.
Thank you very much, sir, for that wonderful and insightful talk. Your valuable words have truly enlightened us and provided us with a deeper
Hello.
understanding of the topic. We are grateful for the time, sir. Thank you very much, sir.
Thank you very much,
Sir,
madam, for giving me opportunity as a resource person in this platform. Thank you, Tejamurthy sir. Hello.
yes, sir.
Hello.
No questions.
Hello.
Any
Is there any
questions, please?
question? Then
Sir, okay, sir. Thank you so much, sir.
thank you very much, madam. Hope I justify the topic.
Yes, sir.
So shall I leave, madam?
Yes, sir, you may leave.
Okay. Thank you, madam. Thank you.
Thank you, sir. So participants, be online. This is the same link you have to join for the another session. Thank you.
Varna madam, can you share the PPT?
Yeah, yes. Is it visible, ma'am?
Yes, ma'am, it's visible. Shall I proceed to your intro, ma'am?
Yeah. Can you please confirm again? Is it visible? Because I changed some settings.
Yeah, it's visible, ma'am. Good
Yeah.
morning,
Okay.
madam. Good morning, everyone. As we move into the final session of today's program, as well as 1 week faculty development program, I warmly welcome all the dignitaries, respected faculty members, and dear participants. I hope all the sessions so far have been informative and engaging. It's now my privilege to introduce and invite our keynote speaker for this final session, Dr. Enduri Sivarna Madam. She is a distinguished expert in the field of pharmaceutical analysis and has made remarkable contributions in the concerned field. Dr. Enduri Suvarna Madam is currently working as Associate Professor, Department of Pharmaceutical Analysis, Sri Anjali College of Pharmacy. Dr. Suvarna Madam is a complete accommodation and research to 12 years of teaching and research experience in pharmaceutical sciences. She specialized in pharmaceutical analysis, instrumental methods of analysis, green analytical chemistry, and sustainability assessment of analytical methods. Her research contributions focus on environmentally sustainable analytical practices, chemometrics, computational modeling, and innovative scopic and chromatographic method development. And Madam has one of the alumni of our Acharya Nagarjuna University. Madam has awarded her Doctor of Philosophy from ANU in the year 20. And as well, Dr. Suvarna Madam has delivered invited lectures and served as resource persons on topics such as advanced strategies and metrics for sustainable analytical practices in pharmaceutical research and green analytical chemistry, the future of sustainable science. And Madam has awarded as Agar Women Excellence Award in the year 2024. And Madam has secured GPAT rank around 442 and NIPER rank at 567 and ANU onset rank of the first rank. And Madam has a lifetime member of APTI. We are absolutely delighted to have Dr. Suvarna Madam with us to share her expertise. On behalf of ANU CPS, I would like to request Dr. Suvarna Madam to kindly deliver the keynote address. Thank you, ma'am. Please proceed with your presentation.
Yeah. Thank you for the nice introduction. Good morning, everyone. I welcome you all to the 1 week faculty development program on next generation drug discovery, leveraging AI tools for advanced pharmaceutical research and regulatory innovation, sponsored by Acharya Nagarjuna University, Rusa 2.0. And as introduced by Madam, I am Dr. Enduri Suvarna, Associate Professor from Department of Pharmaceutical Analysis, Sri Adichunchanagiri College of Pharmacy, Adichunchanagiri University, Karnataka. So today I'm going to discuss the topic analytical sustainability by design from concept to quantifiable impact. So as we all know, analytical method, when we are developing an analytical method, our main aim is to develop a method which must be precise, accurate, or rapid, robust, and it should be selective, specific, and sensitive. So these are the general aims what we are aiming when we are developing the analytical method. But this is the era where we have to aim the method beyond the analytical excellence. That is, we have to concentrate even on the environmental impact. So that is leading to the sustainability. So that is the main concept of this analytical sustainability by design. So we'll see the detailed things in this presentation. So these are the contents I'm going to discuss. Why sustainability? So what is green analytical chemistry and its role? So what are the various metrics with respect to greenness and sustainability? So what is analytical sustainability by design? So case studies and what are the challenges? And finally, conclusion. Hope I am audible, ma'am. Can I proceed?
Yes, you are audible, ma'am.
Yeah, thank you. So what is sustainability and why we need sustainability? So as we all know, sustainability is nothing but use utilization of process or a method for long-term purpose, so without affecting the environment or without depleting our natural resources. So when it comes to the analytical method development, so here the analytical sustainability is nothing but developing an analytical method for intended purpose, and that method should not affect environment, or that method should cause minimum effect to the environment. That is the major aim of this analytical sustainability by design. So these are the different steps, and we all know that. So when we are developing an analytical method, and the major role of this analytical method is, so we can analyze the various samples or wide range of samples like pharmaceutical samples, environmental samples, food samples, or clinical samples. So whatever the sample or the matrices may be, we need a specific analytical method, so for analyzing a suitable analyte. But the approaches what we are generally using are the conventional techniques. So like chromatography, spectroscopy, or the electrochemical methods, or flow injection analysis, whatever. So these analytical methods, generally they are consuming more volume of solvent, and most of the reagents what we are using for the sample preparation or derivatization, so they are toxic. And also the instruments what we are generally using are consuming more energy. So these are the major drawbacks of the conventional analytical methods, but there are the other positive sides also which are giving better sensitivity and all. So but when it comes to the sustainability, so these are the drawbacks of the conventional techniques. So what is the outcome of these drawbacks means? So they are producing more amount of waste. So as we are utilizing large volume of solvent, so definitely more amount of waste will come out. And as if we are using the hazardous reagents, we may get more environmental pollution, so accumulation of toxic compounds in the environment. And as we are using energy intensive instruments, definitely that leads to higher maintenance, and also operator excellence is required. So training of the personnel is also required. So this leads to higher costs. And so how we can minimize it, how we can overcome these disadvantages means by the approach of sustainable analytical practices. So what is the aim of these sustainable analytical practices means? So the major aim of these practices are to reduce the resource utilization and to minimize the waste. So the solvents or the reagents what we are using should be minimal, and because of that, definitely the waste also will be minimized. And also improving the safety. So as we are aiming for using non-toxic compounds, definitely it will minimize the effect to the environment, so that the environment will be safe, and also the operator also will be safe. And then enhancing efficiency. So in the light of this eco-friendly nature or protecting the environment, so there is no compromise of efficiency. We should aim for the analytical performance also. And then how to achieve this sustainability? So there is a integration of green analytical chemistry and also the sustainability metrics in the modern analytical workflow. So we have to merge this green analytical chemistry principles and also some metrics to assess its effect on the environment. So in our regular method development, as we all know, so our regular analytical method development in the present days or the modern days is utilization of chemometrics or analytical quality by design, as Prajapati sir has explained in the earlier session. So these are where we can get better analytical method out of all the trials. So along with that AQBD or the chemometrics, so we can, if we are able to integrate this green analytical chemistry and also assessing its effect during its method development itself, it will definitely give a sustainable analytical method. And here, what do you mean by sustainable analytical method? So as we discussed, so anyway, we are not compromising with the analytical performance. So definitely the analytical performance should be good, like it should be robust, precise, accurate. It should be sensitive, selective, specific and all. And sometimes it might be rapid too. And also, as we are integrating the green analytical chemistry principles, so it should be eco-friendly. So it should have minimal environmental effect. So it should be eco-friendly or ecologically safe. Along with that, there is one more factor called economical. So anyway, we are developing a suitable analytical method or the best analytical method which is not affecting the environment. So in the light of giving or developing a green analytical method or an eco-friendly analytical method, so if we are selecting a costly equipment or the costly chemicals or the reagent, so the final utilizer is not ready to use. The QC labs or the research labs or the academic fields, so they are not ready to use if the reagents or the inputs are costly. So it should be even economic. So as per the sustainability definition, so it should be long-term purpose. It should not affect the environment and it should be user-friendly. It should be easily available. Then only it can be for long-term purpose. So it's a combination of performance, eco-friendly nature and also economic. And coming to analytical sustainability by design. So what is its major aim? So the main aim of this analytical sustainability design is to develop analytical methods, so which are ecologically safe, economically good, and also scientifically robust. So it should be cheap or easily available, and it should be eco-friendly, and also scientifically it should be sound. So then only, so we can prefer it as sustainable analytical method. And what coming to the next thing, so green analytical chemistry and its role. What is green analytical chemistry? So it's a sub-discipline or the subfield of green chemistry, and it's as we all know the green chemistry, which is a very popular one, and it's a sub-discipline of green analytical chemistry, and its main aim is to reduce the environmental impact during the development or the application of analytical procedure. So whatever the analytical procedures we are developing or applying for the determination of analyte, so they should have minimal effect on the environment, because finally the after completion of analysis it is coming out as a waste and it is entering into the environment, so which may affect the environment. So we should minimize that environmental impact when it comes to the analytical method. So its main focus is to reduce the reagent utilization and the use of safer chemicals or non-toxic chemicals and minimal energy consumption. So these are the main pillars or the main aims of green analytical chemistry. So this is the timeline of green analytical chemistry, how it was evolved. So that was the period of 1980s. So where so many research activities were going on. So where the researchers are aimed to analyze the environmental samples majorly. So because of that heavy research workflow, so more amount of waste is coming out of the lab and it is entering into the environment. Again, it is affecting the environment. So there was a sudden ecological paradigm or the ecological shift. So where we need to take the call to protect our environment. So that was the period of 1980s. So then the scientists started thinking about the environmental protection. Yes, we are doing research on the environment, and after all, again, we are dumping waste into the environment, and it is causing the environmental effect. So this is the time where the researchers have to think for alternative research approaches where they can minimize the ecological effect. So because of that thought process, this green chemistry and green analytical chemistry was evolved during 1990s. So the first approach is minimization of solvent utilization. So first they initially started as a clean chemistry or the environmental protection. So then the green chemistry started, and then green analytical chemistry was evolved. So that was the period of 1990s. And then still it got extended as miniaturization and automation. So miniaturization, as we know, we are using the mini devices which require very less amount of the samples, solvents, or everything, the necessary inputs. So miniaturization can reduce the utilization of solvents, and also automation. So automation also will reduce the time, energy, and it will give better results. And then it was a period of 2000s where the metrics were introduced to assess the greenness. Yes, so we are saying that theoretically we are saying we have introduced or we are integrated the principles of green analytical chemistry, and so many methods were developed, which is the best eco-friendly method. So for that, we need some metrics to understand and analyze how extent the method is ecologically safe and which is the best choice among them. So for that, some metrics are introduced. So we'll discuss all those metrics, and then the extension or the advancement in that approach is the integration with AQBD. So as we are developing the best suitable analytical method using the design of experiments or AQBD. So we are developing a good method, but along with that, if green analytical chemistry principles were also introduced into the AQBD, this will give still more better method with more effect, a more eco-friendly effect. And these are the principles of green analytical chemistry. So we have the 12 principles of green analytical chemistry. So the first one is waste minimization. So we need to minimize the waste. So when we can minimize the waste, so when we are using less amount of reagents and salt, so that we can minimize the waste. And then multiple analytical techniques. So if we are able to develop a method which is suitable for determination or the identification of many analytes, so that all those can be determined simultaneously. So for example, caffeine and paracetamol is there. So if we are able to develop a suitable method, a common method for estimation of both the compounds, then it will be easy instead of developing a separate analytical methods. So nowadays, as we know, multi-drug therapy is a preferable therapy. So we need to analyze various analytes at a time so that it will be easy for determination of all those analytes with one method. So multi-analytical techniques are the multi-analyte techniques are preferable. And then energy efficiency. So we should be aimed for utilization of less energy. For example, sample preparation step is there. So if we need to use any extraction methods, heating, cooling and all, and then more preparation is required for the sample, then it is consuming more energy. When it comes to HPLC, we are using ultrasonicator filtration where the energy is also required. So if we are able to minimize such kind of steps, and also if we are able to select or introduce any devices or the analytical approaches where less energy is sufficient, then it will be also a good factor for green analytical chemistry. And then prevention of pollution. So if we are going with selection of green solvents or eco-friendly solvents or the non-toxic solvents and less amount of solvents, all these will reduce the pollution definitely. And safer solvents and reagents. So as we see, everything is correlated with another parameter. So here we have to select stop non-toxic solvents and reagents. And automation, as discussed earlier, automation will reduce the time, energy, and also it will increase the efficiency and minimal sample size. So very small amount of sample should be sufficient for analysis. So that the reagent utilization, solvent utilization will be automatically reduced and automatically the waste will be minimized. And then in situ analysis. The next parameter or the principle is in situ analysis. In situ means on-site analysis. So wherever the sample is available, so we should be able to analyze the sample there itself instead of storing the sample, transporting again, storing in the laboratory, and then analyzing the sample. So it will consume time, energy, and also so many experts are required. So instead of that, if on-site analysis can be performed, for example, you want to analyze water sample at the pond. So if we are able to develop a method or a device where, as we all know, some portable devices are available, so that there itself we can give the result and immediate results, rapid results can be obtained. So it will reduce the risk of transportation, storage, and then maintaining the samples in the laboratory. And then improved selectivity. So we have to develop an analytical method where there should be no interference. So it should be selective to that particular analyte. And then simplification. So the sample preparation or the whole analytical method should be simple. Then only everybody will prefer for this analytic application of this analytical method, and it should be sensitive. That means with low amount or the low concentration itself, the method should give the results and safer disposals. So even after following all these principles, definitely we'll get a minimum amount of waste, and how we are disposing the waste, so that also should be considered. So these are the various principles of green analytical chemistry. So practically it is not possible to follow all the principles, but how extent it is possible, we have to follow and minimize the environmental effect. So what are the various metrics that measure the greenness and sustainability? So we'll see one by one. So there are several metrics available, but I'll discuss the main metrics which give us better understanding what are all these green metrics and how to measure it. So these are the most widely used metrics. First one is NEMI, next one is the modified NEMI, and third one is AGP, and then GAPI and AGRI. So these are the various greenness tools which will measure the effect of analytical method on the environment. So here most of the times we are using the colors which are indicating, like green means eco-friendly and red means it is not eco-friendly. And sometimes we are using yellow or orange color, so which is indicating that moderate ecological impact. So we'll see the details of every metric. The first one is NEMI, so which is nothing but National Environmental Methods Index. So this was introduced by US Environmental Protection Agency. So just to indicate what ingredients or what solvents or reagents we are using, so whether they are causing any effect to the environment or not. So it's a pictogram based. So why we are choosing the pictograms means, by seeing that image itself we can understand in what way it is eco-friendly and what way it is causing toxic to the environment, and how way we can improve the eco-friendly nature of the method. So it is a pictogram based, it is easy for understanding. So we can have the better understanding by just seeing observing the pictogram itself. But the drawback of this is it is not quantitative. So most of the times we may face the situation like we have some 10 analytical methods, what we have to choose. And most of the analytical methods will give us almost the same color. So in that condition, this NEMI is not much suitable, and it is suitable for preliminary screening. So not for in detail understanding, but it is a better method for preliminary understanding and preliminary screening of the existing methods. So here, in case of NEMI, it is consists of 4 quadrants. The first quadrant belongs to PBT chemicals. What do you mean by PBT? It's a persistent, bioaccumulative, and toxic chemicals. So that means, so we have the chemicals list, so which are coming under any of these categories. You know the MSDS data of every compound, material safety data sheet. So that material safety data sheet contain all the details with respect to that particular compound, and if it is coming under the category of persistent or bioaccumulative or toxic, it will be given in that MSDS sheet. So if you refer that MSDS sheet of that compound, so you can understand whether it is coming under the category of any of these three. For example, you are using methanol. So if methanol is coming under any of this category, then you have to keep this quadrant blank. That means it should be left white. If any of the compounds, for example, you are using water, so water is not coming under this category, then this quadrant will be green. So that what it is indicating, you are not using, so as you are using water, you are not using any solvent or the reagent which is not coming under this PBT related category. So that in that perspective or in that aspect, it is safe. And coming to the next quadrant, hazardous waste. So whatever the compounds we are using, if they are coming under the listed waste or not. Okay. Environmental Protection Agency, it has introduced 4 types of waste, that is K, F, P, U. So depending on the what type of source, from which source these wastes are coming. So KFPU related wastes are available and those are listed. So if you search in the Google also, you'll get it. KFPU listed waste. If the compounds or the reagents or solvents, what you are using are coming under this, any of this KFPU listed waste, then you have to consider it as a hazardous. If those are not coming under, the reagent is not coming under this KFPU category, you can consider it as not hazardous. So then if it is not hazardous, you can keep this quadrant as green. And if it is coming under this list, you have to keep this quadrant as black. So then you can understand that, yes, no hazardous wastes are coming out of this method. And then the third quadrant is minimal waste. So we, as the GSE is aiming for minimization of waste, so here some limits were given for this waste. So that is less than 50 gram or ml per sample. So when you are analyzing a sample, what is the amount of waste it is giving out? So there is some limit for that as per NEMI, that is less than 50 grams or 50 ml per sample. So if more than or beyond this limit we are getting the waste, means it should be kept blank. If you are getting less than 50 ml or 50 grams of waste per sample, then it is considered as a green. And then the fourth one is about the corrosive nature. If any of the compounds you are using in the method are strongly acidic or strongly basic, then it is coming under the corrosive category. So that means pH less than 2 or more than 12. For example, HCl. So it is a strong acid. Then we have to consider it as a corrosive. Then this quadrant should be kept blank. If you are not using any corrosive chemicals, then you can consider that it is a green. This quadrant can be green. So these are the 4 parameters or the main 4 parameters considered in case of NEMI. But if we are going in deep, this assessment is not sufficient. So by considering the drawbacks of this NEMI approach, so many metrics were introduced. And here the first drawback what we found was it is very limited and also it is quantitative. So in order to overcome this drawback, so modified NEMI was introduced. So this modified NEMI is a tri-colored one. So like it is dividing each parameter into 3 levels. So like low, moderate, high. So if it is low, that means if the effect is low, it is considered as green. If the ecological effect is moderate, it is showing orange or yellow, that is medium. If the ecological effect is high, it is considered as red. So along with that, some minor changes were made in the NEMI. So like the first one is operator's risk. The second one is energy consumption. third one is waste volume, and fourth one is reagent consumption. So these are the four main parameters considered in this modified naming. So here, what do you mean by operator's risk? So if you observe the chemical bottle, some hazardous pictograms will be provided. Maybe you all know it. So based on the hazardous pictogram, some one word description will be provided on the MSDS sheet. So like hazardous, danger, warning. So one word description will be given. So if it is coming under the category of danger or highly hazardous, then we should consider it as a red. Operator's risk will be high. Okay. So moderately hazardous means, okay, it is medium. So it is not hazardous means you can consider it as a gray. So we can divide it into three categories. And then energy consumption. So here the it is given in the three levels, whether it is consuming more, moderate or less energy. And even for the waste volume also the levels were given, and even the reagent consumption also the levels were given. So it is like, it is not quantitative or it is not qualitative. It is like a semi-quantitative. So we can say whether we can divide that into low, moderate and high, but we cannot exactly give the score. But the advantage compared with NEM is it is covering more parameters and it is divided into three levels instead of saying yes or no. So we can see the different hazardous pictograms: explosive, compressed gas, harmful, flammable, corrosive, health hazard, oxidizing, toxic, environmental hazard. So these are the usual hazardous pictograms we can find on the labels of a chemical compound. So most of the times, if more than two or more than two hazardous pictograms were there on the chemical label, then we usually consider it as a not safe. So it should be two or less than two. If more than two are there, so then we can consider it as relatively not safe. And then the third tool is AGP. So it's a semi-quantitative method. So it is also called as Rani driver tool. So it is a pentagram, so pentagon type of the pictogram, so which is covering four parameters. So it is considering the safety, health, environment, energy, and waste. So it is a tricolor like modified naming. So this is a Rani driver tool, analytical grade profile, and this is a semi-quantitative method, and it is a tricolor, and we can give these colors to each and every parameter, and for every parameter the limits were given. So we'll see that in the table. So this is the table which is giving us the data with respect to every parameter. So as I discussed, so in the MSDS sheet, so the NFPA labeling is also available. I'll show you that NFPA labeling also. So this is the NFPA labeling, which is provided in most of the MSDS sheets. So here, if you observe this diamond, this is a diamond shaped one, which is divided into four categories. So red, blue, yellow, and white. So this blue is with respect to health hazard, red is with respect to fire hazard, yellow is with respect to its reactivity, and white is for specific hazards. And the levels were also given. So 0 to 4 for every parameter, and for specific hazard, some names or codes will be provided. So if the number, you can see the number 0, 1, 2, 3, 4, right? So here, this is for orthophosphoric acid. So where this health hazard is 3, that means extremely danger. With respect to fire hazard, it is just will not burn. And this, with respect to yellow, it is stable. And then when it comes to this special category, it is coming under acid. And then acetonitride, similarly, you can see 230, and it is not coming under any of the specific hazardous category, so it is left black. So this is with respect to NFPA labeling, and you can observe the scores for health hazard and also for fire hazard. Okay, so now coming to this table, so you can get the NFPA health hazard score. So if it is 0 to 1, green, 2 to 3, moderate, so it is yellow, and it is carcinogenic. If it is given as more than 3, it is considered as carcinogenic, then it is red. So this first column is with respect to green, it is yellow, and it is red. So when it comes to safety hazard, the same thing as I shown here, this is the fire hazard. So wherever if the chemical is coming under this category of 0 to 4, we have to find out whether it is red, yellow or green here. So 0 to 1, green, 2 to 2 or 3, yellow, and if it is 4, it is red. And the same thing, the third one is environment. So when it comes to environment, as we are dumping waste into the environment, we have considered amount of chemicals and waste. So here, if we are using less than 50 grams of reagents or the chemicals, then green, 50 to 250 grams moderate, and more than 250 grams of chemicals together, all together, then it is toxic or red. So similarly, low energy methods, they are considered as green as per the principles of GAC. Moderate energy, so they are yellow, and hyphenated techniques which are consuming more energy, they are considered as red. And coming to the waste, similarly, it may be in ml or gram per sample. So for the analysis of one sample, how much amount of waste it is giving. So less than 50, 50 to 250, and 250. So if you are using 250, it is considered as red. So we have to calculate how much amount of chemicals or reagents we are using. And also we have to calculate how much amount of waste it is coming. For example, you are running a sample and the runtime is 10 minutes and you are running with a flow rate of 1 minute. Sorry, 1 ml per minute. So the amount of waste that is coming out is 10 ml. Right? So like that, we have to calculate for the analysis of each sample how much amount is coming. And the next one, it is analytical echo scale. The fourth one is analytical echo scale. It is developed by Galuska et al. In 2012. So in the previous cases, what we have seen are the qualitative type, and also these are pictogram type. So but here this is a quantitative type, and we need to make some calculations, and we can give the score to that to the analytical method. So instead of giving a score, we are subtracting the points from out of 100 and by considering like penalty points. So if we are performing any of our approaches are not ecologically safe, we have to subtract some points like penalty points, and then we have to find out what is the score. So after getting the score out of 100, if we are doing 10 mistakes, means 10 points will be cut off. So like that penalty points will be cut down from this out of 100. And after all, what is the score obtained? So for that score, some levels were given, which is considered as a green effectiveness, and you can call it as a green certificate of the analytical method. So there are certain levels. If the total score is coming in between 90 to 100, we can consider it as category A, dark green. So similarly, if we are getting a score less than 20, means it is not at all ecologically safe. It is like a category G. So this is the tabular column, which is giving us the limits and the procedure how to calculate it. So the first one is amount. How much amount of reagents and what are the reagents we are using. So if the amount is less than 10 ml, we can give 1 penalty point. If the amount of reagents is 10 to 100, 2 penalty points. More than 100, 3 penalty points. So this is with respect to amount or volume. And coming to what type of compound we are using. So as I discussed earlier, so one word description will be given. If that compound is coming under the category of danger or severe hazard, then we have to give 2. Warning or less severe hazard means we have to give 1. If no hazards were there, we have to give 0. So these are the penalty points given for each compound. If we are using 2 to 3 compounds, we have to calculate for all the 2 to 3 compounds. So this is the case where orthophosphoric was used and also acetonitrile was used. So how we have to calculate it? So for acetonitrile, 2 penalty points were considered and it is multiplied with 1. That means 10 to 100 ml of acetonitrile was utilized, and also it is coming under the category of severe hazard or warning. So we have to multiply both, which is giving you 2 penalty points. Similarly, we used OPA, so that for OPA it is 1 penalty point because we have used very less amount, so we are less than 10 ml, and it is coming under the category of warning or less severe hazard. So we have given 1. So totally it is giving us 3 penalty points with respect to this amount and hazard. And then when it comes to technique, how much energy we are using. Like we are using some simple analytical methods or hyphenated techniques. So based on that, we can give the penalty points. So here is the list given. So these are all the methods, titration, IR, spectroscopy and all, which are consuming less than 0.1 kilowatt hour per sample. Then we have to give 1 penalty point. And if we are using atomic absorption spectrophotometer, ICP and GC, LC and all, so it is coming under less than 1.5 kilowatt hour per sample. So we have to give 2 penalty points. If it is like hyphenated techniques and other advanced methods, more than 1.5, we have to give 3 penalty points. So here the example of the method what we have chosen is HPLC. So we have allotted 1 penalty point. And then coming to the occupational hazard, like analytical process hermetization. So if we are performing the method in a sealed area or concealed area, so then the gases or the emissions, whatever coming out of that method, it won't be entering into the environment. If we are following such type of method, then we have to give 0 penalty points. And if any vapors or gases are emitted during that method, so we have to give 3 penalty points. Like any gases are evolved, any leakage chances are there, and during the reaction any vapors are coming out, any nitrogen gas on the reaction it is released, so that and all should be considered. Because it is entering into the environment, we have to give 3 penalty points. And when it comes to waste, so much list is there. If no waste is produced, then it is 0. For example, what do you mean by no waste? So we are just taking the sensor and putting that sensor into the water sample and then removing it, and we are identifying what is the TDS of that water sample. So no waste, right? So then then that is coming under where 0 waste, and we can give 0 penalty points. And if we are getting less than 1 ml of waste, 1 to 2 ml, 3, and more than 10 ml, 5. And then, okay, we are getting waste. What are all the measures we are following for disposing the waste? Are we following a recycling process? Like by distillation, we can regain the methanol, whatever we used in the HPLC, and we can use it for further purpose. Are we following such type of recycling process? So if we are following, then 0 penalty points. We are not following any of the recycling process, directly just disposing the waste, then 3 penalty points. And passivation, that means, for example, acid is utilized. So instead of directly dumping the acid, we have to neutralize that acid and then dumping. Then we can give it as 2. So if any toxic compounds are there, we can convert the toxic compound into non-toxic. So then degradation, 1 penalty point. So like that, with respect to waste treatment, any treatment process are following or no treatment. So for that also penalty points were given. So in this example, we have not taken any treatment. So we have not utilized any treatment. So 3 penalty points. And also 1 to 10 ml of waste is coming out for analysis of each sample. So we have taken 3 penalty points. So all together it is giving 10 penalty points for the analytical method. So out of 100, we have to deduct 10 points. So it is giving you 90 as a score. So as per this green certificate, so 90 score means it is coming under the category of A. So we can consider this method as a green. So as we seen here, so more than 75 means it is excellent. So we can say this one. And 50 to 75 means it is acceptable. Less than 50 means it is coming under red and orange. It is inadequate. So this is not at all eco-friendly. We need to further improve the method with respect to this eco-friendly nature. So for this, we have given this score of 90. So it is coming under the category of A. And next, very, very important tool is Green Analytical Procedure Index, which is in short called as GAPI. So it was introduced by Plotka Vasilka, and it is also a color coded one, similar to the earlier pictograms. So 3 colors will be given, like green, yellow, and red colors, and it is a semi-quantitative tool. So it is not at all completely quantitative, it is a semi-quantitative because it is showing in the different categories of low, moderate, and high. But the main advantage of this GAPI tool is, so it is considering many parameters with respect to each and every step of the analytical method, which were not covered in the earlier tools. So it is considering the 15 plus 1 parameters. We'll see what is this plus 1. So 15 plus 1 parameters, and it is giving a pictogram, which is a group of pentagons. So we know the pentagon as we have seen in the Rani driver tool. So we can see the pentagon. The two, so group of pentagons will be there. So it's a color coded output. Green means high eco-friendly, yellow means moderate, and red means poor eco-friendly. The major disadvantage is it is not giving the score. So that is the main drawback of this GAPI tool. And what are the parameters it is considering? So this GAPI tool is one of the most widely used green metric. So while assessing the greenness of the method. So the first one is, it is divided, as you can see, the 4 pictograms are there outside, and in the middle one more pictogram is there. So totally 5 pentagons are there. So this pictogram is a combination of 5 pentagons. So 4 outer, and in between 1 pentagon is there. So each and every pentagon is discussing about some criteria, like it is with respect to sample handling, and the second pentagon is with respect to sample preparation, and third pentagon is with respect to solvents and compounds, and the last, this pentagon is with respect to instrumentation, and the middle pentagon is with respect to sample, or what type of method we are following. So whether we are directly introducing the sample, or we are storing the sample and we are using any extraction process for the sample, micro extraction or minimal extraction. So based on that, it is given color. And in the middle one circle is there. What it is indicating, as I said, plus 1 parameter. So this circle is indicating the extra parameter, that is with respect to quantification. So whether the method is for qualitative analysis or quantitative analysis. So this is not giving you any score, but this is the circle is indicating your method is for both qualitative and quantitative analysis. If the circle is not there, it is applicable only for qualitative analysis. That means just for identification or interpretation. So we can see the different parameters. So when it comes to the sample handling, it's a collection of sample, whether how you are collecting the sample. So say different options will be given in that, and you have to select the option. So it is an application based tool. You have to download the application and you have to select the options in that application. And it based on the options what you are selecting, it will give you the color. So for this collection, how you are collecting the sample, you have to go to the site and collect, or like that. So it will give you some options, and based on that, it will give you the color. And then preservation, how you are preserving. So whether you need to store it at the room temperature, or you have to store it in the refrigerator, or some other storage conditions are required for preserving the sample. So and based on that, you have to select what is the suitable mode of preservation, and it will give you the color. If no preservation is required, it is considered as green. If more preservation factors are required, storage conditions are required, then higher the preservation factor, it will give you the red. And then the third one is transportation. So whether you need any particular transportation, because if the sample is available near to the lab, then it will be easy. As I said, in situ analysis. So do you require any transportation? No. Wherever the sample is there, you can directly analyze. So for that, it will give you the green color. If any particular transportation is required and you need to use that lab which is very distant, then it will give you the red. So these are the four categories. And then storage. So how to store it, and then the type of the method. So this is with respect to the sample preparation. What type of method you are following? Any sample pre-treatment is required. So with respect to that, it is considering. And then what are all the solvents and reagents we are using? So it is giving us the list, whether it is biodegradable solvent or some of the solvents are biodegradable and we have not at all used biodegradable solvents or reagents, and we have to select that category. So for example, you have used glycerol or ethanol, it is coming under biodegradable solvent. So you can select such kind of option. And then additional treatments. So what are all the additional treatments required? Any derivatization is there? No derivatization is there? And very minimal treatment is there. So you have to select that particular option. And based on that, it will give you green, red and yellow. So if you go on using that application and giving the selecting the option, it will give us colors so that we will get an idea what kind of approach need to be followed in order to reduce the environmental impact. And then next, coming to the solvents and compounds used, 9, 10, 11. How much amount we are using? As we discussed, any health hazard or safety hazard categories are there for that particular compounds. So we have to select that one. So like NFPA labeling, 0 score, 1 or 2 score, 3 score, we have to select. And based on that, it will give you the color. And coming to the last one, with respect to instrumentation, how much energy it is consuming. So as discussed earlier, whether it is consuming 0.1 kilowatt hour per sample or 1.5 or more than 1.5. And then 13th one is occupational hazards. Any hazards are there for the operator or any operator's risk is there. And then coming to the 14th one, waste. How much amount of waste is coming? So it is giving us the options and you just have to click the options. So with certain limits, the options were made and we have to select that option and it will give us the result. And then coming to the waste treatment, so how we are treating the waste. So whether you are disposing the waste directly or you are recycling the waste, passivating the waste. So like that, it is giving you some options and we have to select it. And finally, as I said, so the last circle is whether it is a qualitative or both qualitative and quantitative. So in the previous cases, none of the metrics have considered these many parameters because each and every step is essential for reaching the sustainability or eco-friendly nature. So more parameters are considered means we are assessing the method in detail to reduce the ecological effect. So this is about GAPA. And we can see this overall GAPA pictogram. So when it comes to this sample, how we are treating this, storing the sample, the first category is red. So and here you can see the fourth parameter is yellow. That means it is having moderate effect. Even the sixth one also moderate effect. And 11th one, it is having high ecological effect. 12 and 14, 10 are moderate effect. So by seeing this pictogram, we can get an idea like, so yes, this we have to modify the method in such a way that this first parameter and the 11th parameter should be either yellow or green so that the ecological burden will be reduced. So that is the major advantage of this pictogram. So by seeing the pictogram itself, we can understand so what are all the advantages and disadvantages of this method with respect to eco-friendly nature and how we can alter it. So that is the main advantage of this pictographical representation. And these are the limits given for each parameter. As I discussed, collection of sample. So whether it is inline collection, online collection or offline collection. So inline means you can consider the example of sensors. So it is some reaction is going on in that itself, sensor will be there. Whether the compound is formed, it is completed, that sensor can detect it. You don't need to collect the sample and you need to store. You don't need to store the sample, and we don't need any other instrument. But online or at line means we just have to collect a sample and the laboratory will be beside that sample collection point and you can easily analyze it. And offline means you have to transport the sample. So laboratory is not available there. You have to collect it and we should store it and you should transport it and then it will be analyzed. So that then we can consider it as a offline analysis. Similarly for preservation, so none is required, or chemical or physical is required, like refrigeration and all. So adding just some amount of salt or some other HCl, okay, whether it will be stored or both are required, physical chemical. So based on that, so you can select any of the category. And then transport, any transportation is required. If it is not at all required, we can just put dash and you can do it there itself, means none, and required means it is red. So here only two categories, no moderate, medium category, none are required. So only two. And then coming to the storage, how we are storing. Storage is not at all required, or we can store it in normal conditions or under special storage conditions. So based on that, the color will be allotted. Similarly, what type of sample preparation method, whether you are following extraction, simple procedure, or no preparation, direct analysis. So any direct analysis can be done. And scale of extraction, whether it is nano, micro, or macro. So as we discussed, so miniaturization and minimization of solvent is one of the parameters of GSC. And solvents and reagents used. So what type of reagents we are using? So whether it is green, non-green, or it is not at all using any solvent. So if it is solventless method means it is still better. And additional treatments. Any additional treatment is required for the sample. So for example, you are going to analyze an amino acid and you are treating that with ninhydrin reagent. So sample treatment is required. So whether that process is tedious or easy, that depends on minimal sample treatment or advanced treatment. And amount of reagent or solvent. So it is giving the limits, and based on that we have to select it. And similarly, as I discussed, health hazard and safety hazard, even energy also discussed, and occupational hazard also discussed in the earlier two. And waste also, some limits were given. And with respect to waste treatment, so whether recycling is done or any degradation is done or no treatment. If it is no treatment, we have to consider it as a red. So even you are selecting this particular option of no treatment, automatically that particular quadrant will become red. So this pictographical representation will give us better understanding in what way the method is lagging and how it is causing the ecological effect and how we can further improve the method in the next procedure. And the next most widely used tool is Agree tool. So which is called as analytical greenness tool. And it is a quantitative tool. As we discussed earlier, the major drawback of all the earlier methods is it is not quantitative. So by seeing the colors itself, we can understand how we can improve it, but we cannot say which is the best method. All on the whole, which is the best method, that cannot be known. So that problems were overcome by using this Agree tool. And the main advantage of this Agree tool is for each parameter or for each property or each principle, score will be given, and also the cumulative score will be given for the whole method. So cumulative score and also score for the each method, and the score for the each method is considered, and based on the score it is giving us the colors. So different color grades were there. So you can see 0 to 1 for each parameter, and based on that we can give the we can select the option and it will give the color. So it is also an application based. It's an open source. We can use it as a website, and also you can download the tool and we can implement it. We can apply this green matrix. So it's a 12 sections will be there, and each and every, as we discussed, green analytical chemistry principles are 12. So it was made in accordance with GAC principles. Those 12 principles were included here. So the shortcut for those 12 principles are significance. So by considering the first word of this every principle, the shortcut is given as significance. And here you can say green means same. High greenness, red means poor greenness. In the middle, so many colors are there. So light orange, dark orange, yellow, light green, and then moderate green, and then dark green. So many colors are there, shades were there. And one means it's a green color, completely green color. So you can get a cumulative score, or you can call it as a composite score. So these are the 12 parameters as we discussed in the GAC. It is considering the 12 parameters and it is giving us the options, and as they discussed in the GAPI, it will show us the options, and we have to select the options which is suitable for our method. And even we can give weightage to the each parameter also. And next very important tool is RGB. And when it comes to greenness, this Agree tool and GAPI tools are the most widely used tools. And as we discussed with respect to the sustainability, sustainability is not only coming from the greenness. So it's an amalgamation of analytical performance, eco-friendly nature, and also applicability. Applicability means if it is easily available, if it is economic, then it is easily, you can consider it as practically applicable. Applicability will be good. So by considering this drawbacks of green analytical chemistry or the earlier metrics. So we discussed green analytical chemistry principles of 12. So the 12 principles are majorly focusing or rotating around the greenness of the analytical method and its ecological impact. So it should be environmentally safe. But the environmental safety itself is not sufficient for consideration of the analytical method. We have to consider the analytical performance, ecologically safe, and also its applicability or practicality. So then only this is considered as a sustainable. So the RGB tool is the most widely used tool in order to assess the sustainability of the analytical method. So it was proposed by Paul Novak and et al. In 2019. And not only just introducing the RGB tool, they have introduced 12 principles of white analytical chemistry. So apart from the green analytical chemistry, there is another concept called white analytical chemistry. So it is considering all the factors which are pertaining to sustainability. And based on that, for each parameter, it is giving a score called color score, which is considered as 0 to 100%. So it is one of the major tools considering for sustainable development. And it is not an open source tool or app based tool. It is just an Excel template, which is very easy for operating. And this is the RGB algorithm model developed by Paul Novak et al. And they have considered red color for analytical performance, green color for eco-friendly nature, and blue color for practical applicability. So if the method is red means it has only analytical performance. If the method is green means it has only eco-friendly nature. If it is blue means it has only practical applicability or it is economic. But what we want for the sustainable analytical method, we want a combination of these three. So if the method developed is combination of these three, these three are primary colors. So these three primary colors together are giving us our ideal white color. So if we are able to develop a method which is a combination of all the three, so then it is considered as a white method, which is a sustainable analytical method. And what are all these three colors? These are secondary colors, so which is a combination of these two, red and green. Yellow is a combination of red and green. And cyan is a combination of green and blue. And magenta is a combination of red and blue. So what these secondary colors are indicating? So after getting the score, if your method is showing yellow color, that means it is just analytically good and also ecologically safe, but it is not its applicability is not good. So it's a combination of red and green. And the final score, what it is showing is magenta color. That means it is analytically good and practically it is good, but it is not ecologically safe. And if it is cyan color means its analytical performance is not good, but it is good in eco-friendly nature and productivity or practicality. So in order to manage or equilibrate all the three, so we need to consider all the parameters, then only we can achieve sustainability in the analytical method. Then we call it as a white analytical method. So if we are getting any secondary colors means, so there is some drawback in the analytical method where some improvements are required. So this is the Excel sheet, and in the supplementary file, the Excel sheet is provided, and this is the overall look of the RGB model, and we have to just give the scores in this called cells. So in the Excel sheet, in the cells, we have to give the score of this age parameter. So what are they? So these are the 12 principles of white analytical chemistry. So and those 12 principles were divided as R, G, B. So 4 principles with respect to R, 4 with respect to G, and 4 with respect to B. So when it comes to R means red, that means analytical performance. What is the scope of application? How extent it is sensitive? So with what minimal concentration we can utilize that method. So for that we can give the score, and LOD and LOQ, what are all its values, and coming to precision, how extent it is precise and what is its accuracy. So for all these, every parameter, so the limits were provided in the supplementary table, supplementary sheet. You can please refer that sheet, and based on that we can give the score for our analytical method. If you are just inputting these values here, it will directly give you what is the score with respect to red. And then you have to give this one, when toxic reagents were used, what is its toxicity. So for that also limits were given, and you have to refer the supplementary file, and based on that you should give the score, and what is the amount of reagent and waste produced, and for this also limits were available, and what is the energy of the instrument or the other ancillary equipment we have used. And then the fourth one, G4, is direct impact. What are all the effects on the environment? And based on that also we have to give the score. It will directly give you here. And then when it comes to practical applicability, till now we have not discussed this type of criteria. So cost efficiency, what is the cost? Because as we discussed, if it is costly and easily not available, so we cannot prefer this method. So what is its cost, and what is the time efficiency, with whether we can run it in a real sample analysis, so whether we can make it fast or not, like that it is considering the parameters. And when it comes, the third one is requirements. What are all the requirements to maintain that equipment and all, and what is the operational simplicity, whether any trained personnel is required, and what are all the safety measures he need to take. So all those will be considered in case of operational simplicity. And any AC is required for for maintaining that equipment. So what type of laboratory conditions are required. So all these will be considered in case of this practical applicability. How extent it is easy, how extent it is simple, it will get good score for practical applicability. So anyway, it is giving score for each and every category of R, G, and B, and based on that, accumulative score will be given here. So as you can see, the white color. So that means in all the 3 categories, it is good, and so it is showing white. So if the score is reduced, and from what perspective the score is reduced, that will be considered, and based on that, the color of the column will get changed. So as we discussed here, if the overall method it is showing yellow color, so then we can consider that it is not practically good. It is only eco-friendly, and analytical performance is good. So we have to select the method based on your intended purpose. Okay, you have very good resources, you have all the reagents available, but
Madam, your voice is not audible.
Is it visible? Ma'am, is it visible?
Yes, madam. Screen is visible, ma'am.
Yeah, thank you. So we are discussing about the RGB model. So based on the color score for every category, and finally we are getting a score, whiteness score for the overall method. So which gives us an idea in what way the method is good and what are all the drawbacks of the analytical method. So if your requirement is only with respect to the performance and eco-friendly nature, so then we can go with the score with respect to this yellow color. And if your perspective is not with respect to greenness, it is with respect to practical applicability and only performance, means if you are getting magenta color, then also you can go with that one. So it depends on what is the final requirement from your side. And then these are all the colors given and what are all the scores, how it is changing the color. So there are some limit for the scores. So more than 66.6, in between 66 to 33.3, and more than 66. And based on that, it will give us the color to the method. So if it is more than 66.6 means it will give you good results. So it is satisfactory. So if it is in between 66.6 to 33 means so moderate, and if it is less than 33.3 means it is not satisfactory with respect to that particular aspect or the parameter. So then we have to improve the method based on the aim. Then, so till now we discussed about what is green analytical chemistry and what are all the metrics with respect to greenness and also with respect to sustainability. Now, how to integrate that greenness into your analytical method. So we'll see that one. So what is analytical sustainability by design? So actually, whatever the approaches we discuss, the conventionally the method what we are following is, so we are developing an analytical method, and after optimizing the developer method and after application, then we are assessing its environmental effect by application of any of the metrics. So you can see different publications. So we are developing a method, and then after optimization and application, so we are assessing the environmental effect of the analytical method. So that's what the usual approach what we are following for the method in the method development. But what this analytical sustainability by design is saying, so it is saying to incorporate the principles of green analytical chemistry from the step of optimization itself. When we started designing the method, so we have to include GAC principles into the our aims, into our objectives. So then only we can have that idea of GAC, and we should optimize the method by considering even the green metrics also. For example, as sir discussed earlier, we are going with AQVD process, box Benham design, and based on that we are giving certain levels as we want to minimize the tailing factor, or we want to increase the resolution, and so we want to decrease the runtime. So we are giving such aims and we are developing the method. So along with that aims, we have to include that, yes, as per the green metrics, we have to receive this much of score for our analytical method. So if your approach is like a proactive approach, then instead of just assessing the method after optimization, so you will be including that GAC during the method development, and you will definitely get a better method without any compromise in its analytical performance also. So we have to use quantitative green metrics for evaluation, and then when you are incorporating all these things during its design itself, definitely it will be balancing the environmental parameters, economic parameters, and also operational factors. So all will be balanced, and then it will ensure that it is efficient, the method is safe, and it is ecologically responsible analytical method. So the main aim of this analytical sustainability by design is to develop analytical processes that achieve high performance with minimal environmental impact, so that But during the design itself, we are incorporating all these criteria, then it will definitely give us the best method without any compromise in its analytical performance or practicality. And these are the 5 pillars or the 5 main approaches for sustainability. So what are the 5? When we want to achieve sustainability means if we are following these 5 strategies, it will give us the best results. Miniaturized techniques. When sample preparation is there, instead of going with conventional extraction techniques, we'll go with micro extraction or lab on chip or some portable extraction devices. And then alternative detection system. Instead of going with the heavy instruments or the hyphenated instruments, if you are able to develop a method with portable instruments itself with better sensitivity and accuracy, and also the intended aim of the analytical method also should be considerable. So like smartphone based colorimetry, portable spectrometers, so like that. So we can choose alternative detection systems. As I discussed, for water analysis, we don't need any heavy instruments. So small devices are sufficient for analyzing the water samples. When it comes to the ion analysis, total carbon content or total dissolved solids, that and all, so heavy instruments are not required. And then selection of green solvents, so which are biodegradable, which are easily available, and it should not cause any toxicity to the environment. So this is one of the important approaches. And also waste management. So if instead of treating the waste, if we are able to develop the method which is using less amount of sample, less amount of solvent, or solventless method also can be preferred. So that will give us very less amount of waste. And then energy reduction. So this alternative detection systems and this energy reduction are correlated with each other. So we have to use the less energy required instruments, and also the sample preparations or storage conditions what we are using should not consume more amount of energy. So that is also one of the essential things with respect to energy reduction. So if we are following these strategies, then we can easily achieve the ecologically safe or environmentally strong method. And then, so as I discussed, so integration of green analytical chemistry with AQBD, so that will give us the analytical sustainability by design. So during the method itself, we have to integrate these principles. So as we know, AQBD is majorly aiming for robust, reliable, and efficient analytical method. And we are in that, along with that, we have to integrate optimized solvent selection. So what type of solvent should be selected? So generally what we are preferring, so acetonitrate, and we'll change the buffer and we'll change the ratio. So instead of that, any other alternatives are available to develop an analytical method, so which is giving good resolution, so good response, which is ecologically safe. So that parameter should be considered during the optimization itself. So then reduced experiment. As we know, AQBD will reduce the experimental trials and will, so though initially the trials will be more, but still it will give us the trials in a proper manner. And enhanced method life cycle management, as Sir has discussed, and we have to integrate GSE principles during the method optimization itself. Then it will be easy for us to get the ecologically safe method and sustainable method. So this is the ASBD workflow framework. So you can see, so as in the AQBD, we have to first identify the problem. So what is the problem identified? Maybe the tiling factor, maybe the resolution, or maybe the retention time or the run time. So we have to consider the problem identification. Along with that, we have to consider based on the literature review, we should consider that yes, in the previous literature or the existing methods, so toxic solvents were used. So your problem should include with respect to green analytical chemistry also. Maybe some sample preparation procedures are tedious. We have to consider that also as a problem identification. And then optimization of the analytical technique. So by using the UQBD, so we have to select the analytical parameters, as you know, critical quality attributes, critical method parameters and all will be considered. And then green solvent and reagent selection. So how to select this green solvent and reagent? So many companies like GSK solvent selection tool. So many companies are providing this solvent list, which is a green solvent, which is affecting the environment, which is a dangerous solvent, and which is a banned solvent. So the list was already there, and along with that, the recently some tools were also introduced with respect to solvent selection. So like green solvent selection tool, GSST tool is available, and it is giving us the score with respect to greenness. And if you select the any particular solvent, it will give us the idea which is relatively ecologically safe. And based on that also you can choose. And even it is providing the log P values, what is its polarity and all. So because we need to know the polarity values and all while selecting the solvent. And then method optimization. So you know MODR will be obtained, method operable design region. So based on that we can optimize which is required for us. And then assessing the sustainability of the method using metrics. So as here we are aware about the selection of the solvent and minimizing the reagent utilization and solvent utilization, definitely that will show the good score in the metrics of the optimization of the method. And then implementation of the analytical method. So for what purpose we develop the method, then we can implement it. So usually what we are following is identification, selection of analytical technique, method optimization, then implementation, and then metrics, assessment of the method with metrics. So that is the usual procedure what we are following. But what ASBD is suggesting is you have to integrate green solvent selection. So during the selection of analytical parameters itself, and you should assess the method before application of the method, so that if in any parameters, if it is lagging, we can still optimize it, and then we can implement the method of our intended purpose. So that is the workflow with respect to ASPID. And we have several pictograms or the tools available in order to assess it. And as I discussed, spider charts are available, green solvent selection tools are available in order to select a suitable green solvent which is ecologically safe. And you can even assess the parameters after assessing the method by using these metrics, and based on the color score given to the each and every parameter, you can understand in what way it is not ecologically safe, and are there any chances to improve that particular parameter, that can also be considered. And then you can re-optimize the method, and then you can implement the method. So that is the with respect to analytical sustainability by design. And coming to the case studies, so we have developed so many methods, and we have assessed so many reported analytical methods with respect to its greenness and sustainability. I think as we are lagging with time, I'll just move a little bit faster. So this is the article published in IJPER. So where the greenness of the UV spectroscopy methods were analyzed with respect to amlodipine bacillate and telmisartan. So these are the scores obtained when we have analyzed the method with NEMI, Rani driver tool, and analytical echo scale and agree. So as per this, so this is the method one, the first method which has used methanol. It is showing relatively ecologically safe, because in the next case, the HCl and NaOH were used, which is reducing this greenness. So like that, so some metrics, some green metrics were used to assess the greenness, and we found this first method, where methanol is used, was relatively safer. And then another one, so the same amlodipine distillate and telmisartan methods were assessed for its sustainability with respect to RGB model. And here, this score and the GAPI was also performed. So as we discussed, GAPI score will give us more information, but for comparison, it is very difficult. So here you can see so many red regions, and it is very difficult to compare which method is good. So we have to count how many red regions, how many green regions are there. So it is little bit tedious process. So by considering this disadvantages of GAPI, a new modified tool was introduced for GAPI, that is called modified GAPI. Along with the colors at the down, it will give us the score also. So nowadays they are introduced modified GAPI. Just for your understanding, whatever the tool used earlier, which will give us the best picture, I have discussed that particular tool. But nowadays modified GAPI is also available, which is giving us the color score. Okay, and then RGB model was used for assessing the sustainability, and here you can see the method 2 is showing relatively good color, good whiteness, and here all the analytical parameters are good. And with respect to practicality, here it is little bit less, you can see that one. And here with respect to green, second amount of reagents, it is more. And also with direct impact, it is scoring less. So because of that, this greenness score is relatively less, and it is showing only the 70 score. If it is 80 or more than 80, it will be more good. And but here it is not passing that more than 66%, and here also it is relatively less. But overall, the better sustainable method is this method 2 with respect to this RGB model. So that's how we can understand, and even by seeing this, we can understand that we can develop a method where we can use less amount of reagent and where it is producing less waste, and where we have to choose a solvent which has less impact on the environment. And also we should choose a method which is simple in application. So if we are able to develop method by considering these parameters, then definitely the score will be improved and we can give a better sustainable analytical method. So that's how we can assess this metrics. And then another work with respect to this. This is a spectrophotometric approach. So for simultaneous determination of amlodipine bislate and telmisartan. So here, as I discussed, we have used this AS by D approach because the methods, whatever reported earlier, have used acetonitril, methanol and all. So, but we have used propylene glycol. So, which is relatively ecologically safe, and we came out with a very good analytical performance, and also it gave so ecologically safe method. We can see the score of this method, and here the sustainability also it has improved to 81.3, and this is another type of tool called BAGI. So blue applicability grade index, which is with respect to only applicability. So like this, so many tools were there, but what are all the most widely used tools that were discussed by me? And this is another method which is published in the scientific reports. So this is a green IR method. So for quantification of amlodipine bislate and telmisartan. So because as we are not at all using any solvents except the eco-friendly KBR, so it was found to be relatively ecologically safe, and it will have given us the good results also. So you can see the method, whatever the reported HPLC method and the proposed FTIR method, it has given good results. So this is a modified GAPI, it has given the score also. And this is AgriPrep. AgriPrep is the advanced version of Agri, and it is considering the sample preparation parameters, so extra parameters also. And the next one is RGB model, as I discussed. So the method what we developed, it has shown 87.2, so relatively more green when compared with the existed or reported HPLC method. And the next one is, so this is another assessment. So where the antihypertensive amlodipine, telmisartan and hydrochlorothiazide were assessed. So you can see the drawback of NEMI here. Most of the pictograms are giving white color and remaining all are green. So how we can say which is relatively economically safe? So that is the drawback of this. And that's why the quantitative metrics are highly preferable. You can see the scores here. So with respect to ESA, the first method is good. So with respect to AgriPrep, this is good. And with respect to modified GAPI, so the second method is relatively good. And with respect to this RGB fast, this is a more good method, that is 62, the method 3. So if we are preferring for sustainable method, you have to go with RGB fast. If you are going with only greenness, so you can go with GAPI and Agri. And AgriPrep is preferable when you are having a tedious sample preparation, then you have to go with Agri. And then these are the preliminary tools which were not much preferable now. So this is published earlier. And then this is digital image colorimetry method, where I discussed that portable devices can also be considered. So here we have applied this agree tool, where we got 0.74 as a agree score for estimation of isoniazid using this digital image colorimetry, that is using smartphone based application. And we got the good score, and it was published in Future Journal of Pharmaceutical Sciences. And this is another new method. So for tetracycline and tinidazole, so no method was developed till date. So we have developed a new analytical method. So with water as a solvent, and we got a good results with respect to analytical performance, and also the greenness score was found to be good, and also the sustainability was found to be good, as we have not gone with hyphenated techniques. Practical applicability score is also found to be good. And then the recent one, so which is published in BMC chemistry. So like for the simultaneous determination of three antihypertensive drugs. So here we have used another approach, another discussed approach, like analytical sustainability by design. So along with AQBD, so we have considered the green solvent selection tool for selection of the solvent, and we have aimed for reducing the solvent utilization by reducing the runtime. So by using this gradient illusion method, so relatively we got very good score than the existed analytical methods, and it was found to be ecologically friendly, and also it was found to be sustainable. And this is another article published in Critical Reviews in Analytical Chemistry. So with respect to the sample preparation, so there is another tool called SPMS tool. If we are going with analysis of only sample preparation technique or the extraction techniques, then SPMS is the best tool. And then this is the article published 2 years back in RJPT. So this article gives us the idea with respect to all the metrics that were there till date then, and how to utilize it. Because whenever we are learning all these green metrics, we found so many drawbacks and so many points were missing while explanation. So with the thought of make the users understand it easily and with a detailed approach, so we have published this review article on all the green metrics available by the time, and it is giving us a clear picture and the step-by-step procedure how to execute every green metrics, every tool available by that time. And this is another approach where the sustainability of the analytical methods were assessed using the RGB model, and it was published in elsewhere Q1 journal, that is Green Analytical Chemistry. And this is the recent one, so which is published in Green Analytical Chemistry. So that is integrating Green Analytical Chemistry into higher education using a certificate model. In 2023, our university has conducted a certificate course on Green Analytical Chemistry, which is a credit course, and it is of 31 hours. So which was held during January to March 2023. It's an online course. So for that, elsewhere has given recognition stating that this is the first certificate model in Karnataka and also probably in India. So which has conducted a certificate course with respect to the awareness on green analytical chemistry. So for that, we received a recognition as a publication part. And coming to the challenges, so there are several challenges because we need to consider more greenness parameters, and sometimes maybe we need to compromise with the analytical performance when we are in the light of greenness. So there are some practical challenges will be there. And green reagents, so we have very less green reagents availability. We need to explore more reagents and also more solvents, so then only we have the choices of selecting the green solvents. And definitely some high initial inputs will be more. So in order to purchase this miniaturized instruments and all, so we need to have some initial cost should be, investment should be there. So these are the challenges. And coming to the future perspectives, until now there is no integration of AI in this metrics development. So we can even develop tools where the AI integration is also there. So there is a lot of scope for AI integration in this metrics also, and greener mobile based sensors. So we have to develop sensors also where the where no solvents are required, which is considered as a greener sensors. And also, as we know that sustainable development goals, so these green, if you are following these GAC principles in your method development, so it is contributing to the SDG 3, which is good health and well-being, sustainable cities and communities, and also responsible consumption and production. So it is aiming for even the SDG goals too. And finally, the conclusion is, so this ASYD approach, analytical sustainability by design approach, it is a proactive approach, and which is integrating the principles of green analytical chemistry into the analytical method development in an advanced way. And we need to integrate green analytical chemistry, design of expert like IQBD approach, and also the green metrics while optimizing the analytical method, so which will give us the best results. And what is the strategic pathway means, this, if you are following this pathway, definitely we'll get robust, ecologically safe and cost effective method, and which will be giving the result of sustainable analytical method, and it is aligning with even the SDG goals also. So these are the references I used. So just last words. So it's a time to shift from being a consumers to the caretakers, and we have the power to shape sustainable world through our choice. So we have to select our choice, which should be ecologically safe, how we can protect our environment. Thank you. So if you have any queries, you are open to ask. Ma'am, mine is finished. My presentation is finished.
Thank you, ma'am, for a very informative and insightful session. Any queries from audience and participants? Okay, your valuable expertise have truly enriched our knowledge, ma'am, especially on green analytical techniques and RGB assessment model. I'm sure all the participants have gained useful insights from this wonderful talk. With this, we conclude the final session of the 1 week faculty development program. Now we will be shortly proceed to the online posted in the chat box, followed by valedictory session. I request all the participants and dignitaries to stay back and join us for the valedictory session. Thank you. Dear participants, already link will be posted in the group. Please check it and do the online test.